Okay, so here's slide 12, looking at the thin and thick filaments and the proteins that make up them. The thick filament is basically one uh, protein called myosin, but it's pretty complex. Each myosin molecule consists of this long tail of two twisted fibers, plus a couple of heads here with actin binding sites and ATP and ATPase binding sites on what's called the heads, or sometimes also called the myosin cross bridges. They are going to be actually attaching to actin in the thin filament. Thin filament is much more complex in terms of how many proteins there are. The major protein is called actin. And there's these red balls right along here. The tropomyosin is these long green filaments here, and every once in a while you see a complex of several proteins that's collectively called troponin, and there's a calcium binding site on troponin, and calcium is the trigger for muscular contraction. Okay, on slide 13 here, we're going to see how the two types of filaments, the thin and thick filaments, are arranged in skeletal muscle in what are called sarcomeres. And the sarcomeres are what gives the um, skeletal muscle a striated appearance. And there's sarcomeres also in cardiac muscle, but they're a little different, but they're very similar. And um, and you can only see actually sarcomeres only in longitudinal section. We're going to see down here in the lower right that if you look at skeletal muscle in cross section looks very different. So down here lower left is a sarcomere, and it's the smallest um, part of a muscle that actually contracts. And you can see that the thick filaments intersperse between thin filaments. And sarcomere exists from what's called a Z disc to a Z disc, and there are zones. There's or there are bands, an I band interspersed with A bands, and that's what really makes up the striations, and there's an H zone in the middle. And you can see that at the M line in the very middle, you can see just myosin, and the myosin are actually connected to each other by accessory proteins. H zone is just thick filaments. I band is both thick and thin filaments, and you'll see that every thick filament is surrounded by six thin filaments in a hexagonal arrangement. When you get to the I band, you'll just see thin filaments with these elastic uh, proteins called connectins, and then in the Z disc, you'll see those two plus some accessory proteins to hold things together. Slide 14, let's look at other structural and functional proteins. As we mentioned, connectin or titin. Those are elastic, allow the sarcomeres to pull apart once the contraction ends. And we're going to learn how contraction starts, and it's also important to learn how contraction ends. There's something called nebulin, which allows the orderly construction of sarcomeres, and there's one called dystrophin, which anchors myofilaments to the sarcolemma and is the protein that is defective in the genetic disease called muscular dystrophy. Okay, mitochondria and other structures. We're going to see that skeletal muscle is there are different types of skeletal muscle. Some have more mitochondria than others. When we get into those, we'll talk more about that. But there are usually lots of mitochondria per mesial cell. There's myoglobin, which is this oxygen binding protein that's found in skeletal muscle, and something called creatine phosphate, which is a really quick source of ATP. It's not really storage of ATP. You can't store ATP, but it's the next best thing, and it's the quickest source of ATP. We'll get into more of that when we talk about skeletal muscle metabolism. Here's slide 16. We talk about the nerve-muscle relationship, motor neurons and motor units. So the axons of motor neurons are called somatic motor fibers, and they're voluntary. You have voluntary control over most of the time. There are certain instances where you don't. We'll talk about those later on. A motor unit is all the muscle cells that are innervated by the same motor neuron. Size is variable, small in intrinsic eye muscles, large in calf muscles, and they work in shifts. And we've already actually talked about this. We talked about the motor homunculus in the precentral gyrus, how the motor units are very, very small in small muscles control your fingers, your face, eye muscles, very large in like back muscles. And remember, skeletal muscle cells are slaves to the motor neuron. They don't do anything unless they're told to. They're stimulated by their motor neuron. Um, and and then we'll look at the motor muscular junction. This will be in the next video, also called the motor end plate. And they're all the same. They all use acetylcholine as a neurotransmitter, and they're all excitatory. And so we'll look at the motor end plate, the postsynaptic membrane. So the presynaptic membrane is actually nerve tissue. The postsynaptic membrane is sarcolemma, is muscle cell membrane. Okay, slide 16 shows you two motor units here. The red motor units and the blue one. And they're innervated by different motor neurons. And it's important to realize that every single muscle cell is electrically isolated from every other muscle cell. So if the red motor unit is turned on, those red ones will fire, the blue ones will not, even though they're right next door, because they're innervated by a different motor neuron and are a different motor unit. And the strength of a muscle is determined by how many motor units you turn on. If you just have to make a small amount of force, you just turn on a few. And if you have to cause more tension, you turn on more and more. And if you hold that tension, motor units will cycle on and off to prevent fatigue. But of course, fatigue will eventually happen. So it's important to realize the importance of motor units.